The first company is Angel City Bio and Wael. Excellent. Hey, Wael. Thanks. Nice to meet you in person. Here you go. Yep.
All right. So it's 3 minutes, so I decided to do something a little different. So Angel City Bio develops drugs that are targeted to the intestine and liver. We have a platform, but it's not AI enabled. But this slide is AI enabled. And so it's really hard to express in words what our patients experience in clinic. And so I asked DALL-E for some help depicting that with abstract art. And I imagine that the AI algorithm scraped the web and picked up some themes, like the alienation of not being able to join a meal with your friends and family, or picking up a prescription knowing that it's not going to save you, or tragically losing a pregnancy in the 3rd trimester. And as clinicians, everyone on our team is a clinician, we have this privileged view, but we also have strong backgrounds in basic science, clinical research, and operations. I don't have time to go through to tell you how amazing my team members are, but just know that we've achieved at high levels in our respective fields, and we are unshakably committed to our mission. I mentioned we're a platform, but we have a lead candidate, and that's for Crohn's disease. Crohn's is a form of inflammatory bowel disease. You can think of it as an autoimmune disease of the gut, and it arises in young adults, and it's chronic and lifelong. There are different forms depending on the location and the severity. The most common site is in the ileum. That's the small intestine where it meets the colon. While mild disease makes up half of cases, there are no FDA-approved therapies for the maintenance of mild to moderate Crohn's. Indeed, all of the commercials you see for Crohn's disease are for therapies for severe cases. And you can imagine seeing a patient in clinic, a young woman contemplating pregnancy, gets diagnosed with mild to moderate Crohn's. It's a really hard sell to get them to take a biologic with all its associated side effects. If you remember one slide, let it be this one. And so with traditional drugs, whether they're given orally or through IV, they get distributed off target. This gives rise to side effects as well as decreased efficacy. The main crux of our innovation is that we keep the drug within the digestive system. This improves the efficacy and reduces side effects. We do this by conjugating or connecting the drug to a bile acid. And this allows it to go through the circulation of the bile to the intestine and back to the liver. This circulation happens 12 times a day, and so that we get 12 shots on goal. Maybe during Q&A we can dive deep into the data, but effectively what we've shown is that the drug goes where we thought it would go, and it is effective. Whenever you have a targeted therapy, you are by definition narrowing your market. But even with mild to moderate Crohn's ileitis, it is still a very large market. Regarding IP, we have filed our initial patent through UCLA, and it's exclusively licensed to us. It's under review. We also have a second provisional patent drafted. Other candidates include a treatment for intrahepatic cholestasis of pregnancy. This is the most common liver disease of pregnancy. It can lead to the loss of the fetus in the 3rd trimester, and there's been no innovation in half a century. And our other candidate is for cholangiocarcinoma. This is bile duct cancer. So we think we're very well suited for this type of cancer. And it has a worse prognosis than pancreatic cancer, and there's no effective therapy. So I've told you what the world looks like for our patients now, but what will the world look like if Angel City Bio is successful for our patients? And the truth is we need a partner to get there. Thank you very much.
How long it's going to take you to take your product into the clinic?
So, the truth is, I don't think Angel City Bio will take the product all the way to that goal. We envision getting through to human trials phase 1, 1B probably, and then licensing out to a larger pharma company. If everything goes swimmingly, that might take about 3 to 4 years.
Mm-hmm. And who would be a target for acquisition or a partnership with Angel City Bio?
So, I I think pharma companies like Takeda would be great. You know, they are very strong in GI. Or you could look at some of the incumbents that have a lot of drugs already approved, like AbbVie would be another player to consider.
Whose drug are you using for ABC1?
Yeah. So for this one, it's, you know, we use something called mesalamine. It's a drug that's been around for a long time. It's a topical anti-inflammatory medication, and it has a very strong safety record. But it has been difficult to target this drug in Crohn's, and historically that's been a big challenge for its use in the past.
I may have missed it, but is this orally dosed?
Yes, it's orally dosed, so that's a huge advantage.
Okay, and are there any, so presumably it has to be a small molecule that you're conjugating?
Yes.
Have you tried any other types of molecule?
So no, so we, you know, at this point we have self-funded, and so we've only had this candidate tested.
Wow, can you just, what kind of studies have you completed? Were those in vivo or in vitro?
Yeah. So this study was a pharmacokinetic study. We used rats. We used rats because we could stick a catheter in the bile duct and remove the bile and measure the concentrations of the drugs we gave them orally. And so this shows that the drug is highly concentrated in the bile. There's minimal absorption into the blood, and that there's signals that it is indeed undergoing that circulation multiple times a day. And then the other study we did was an efficacy trial in mice, and it was a chemical model of intestinal inflammation. And one group of mice got the intestinal inflammation but a control, but a placebo, and then the other group of mice additionally got our drug, and we saw a huge difference in the amount of inflammation in the intestine. And whether or not, you know, you have a science background, you can see that the intestine that got treated, the projections are longer and they're thinner, and there's not as much thickness in the pink areas, which is like the muscles. And so it was drastically improved. In our cohort, it was pretty universal. Of all the mice that we treated, they had an improvement.
So I could imagine that the side effect profile is decreased, but what are the side effects anyway that you would imagine with the therapy?
So, you know, in order to answer that question, you know, we would have to do thorough toxicology experiments, which would be in the plans. But I can tell you that in our efficacy trial, it was a 10-day treatment. All the mice did well. If anything, they had much better appearance compared to the mice that got the control, and that's a good sign. But and also knowing that the two agents we're conjugating together have a history of being used individually, therapeutically, and have a very good safety record as well.
Okay, thanks.
What does your competitive landscape look like if this is rather has been done before with the other molecules?
1 second. Did I put this in here? Well, yes. So there's two main classes currently for the treatment of IBD. And again, all of these are for severe cases. There's the biologics and there's newer small molecules. The biologics are all parenteral, meaning IV or injections. They're all immunosuppressive. They're probably safe in pregnancy, and they all have really disappointing efficacy rates. Even though, you know, they sound like they're very, you know, effective, they're 20 to 30% actually going to get to goal. And so with the small molecules, these are second line because they have black box warnings associated with them, including blood clots and cardiac side effects. The etrasimod hasn't been released yet for Crohn's, but it has cardiac side effects as well. And they're not used in pregnancy because it's, you know, not clear if they're safe. And again, their efficacy is pretty low too. And again, these are all for severe.
I guess my question is, why can't someone else just develop a conjugating molecule?
So I think the best way to answer this is an analogy. So there has been attempts at conjugating drugs to bile acids in the past. They haven't been very successful. And I think our key innovation is actually the formulation and the drug delivery vehicle. And I think you can make comparisons to something like Moderna or BioNTech. mRNA is a naturally occurring molecule. That's not the innovation. Like people, like using it therapeutically, the idea to do that has been around for decades. The real innovation is when they figured out how to get the mRNA into the cells. And so for us, it's how do we formulate it so that it gets absorbed appropriately at the right place. And so that's the secret sauce.
And how much money are you raising for
Yeah,
what goals?
At this point, we're pretty early on. We're looking to do further studies. We're raising about $500K.
How long does it take you?
Excuse me?
How far does it take you?
So the milestones that we envision this will take us through would be to do an efficacy trial using a genetic model of Crohn's ileitis, as well as rounding out our pharmacokinetics. So we just did basic PK studies. And if we have extra funds after that, we would start building up some of our other drugs in the pipeline.
Is the API that you're using in ABC1, is that in the UCLA or the licensed IP, the foundational IP that?
Yes.
And have you done any significant optimization of the sort of conjugated molecule itself, either the bile acid side or any changes to the API?
So we haven't, because for us, we did a lot of planning up front to try to to estimate what would be the best site of conjugation. But due to funds, we couldn't just go through every permutation. That's something we could consider doing, but we got such a good result with this one that I'm not super compelled. I don't think that's our next step necessarily. We could maybe down the line try to do that to enhance our IP, for example, or extend it. Yeah.
Do you think your secret sauce, as you explained it, would have to be adapted for other APIs?
Not really. No. Good.